Hello everyone, my name is Sandeep Singh and today I will be talking to you about the geographic prevalence of Lyme disease while considering factors like host choices, host seeking behavior and tick density. So to kind of get into overall topic, we have to go into specifics of Lyme disease. So Lyme disease is caused by spirochaete called Borrelia burgdorferi and includes a ton of different types of species. So a spirochaete is constantly in motion. It has a corkscrew motility that helps it disseminate into tissues and burrow into tissues. There is also a worldwide distribution of Lyme disease, primarily in Asia, Europe, and North America. And Lyme disease is transmitted by ticks, specifically in the US. And think about exotic scapularis and exotic pacificus. For this case, we are going to be talking about exotic scapularis, some signs and symptoms of Lyme disease, including arthritis, neurological conditions, bull's eye rash, fatigue, fever, headache, aches and pains, and even more that are not as well understood. There is also a few different disease manifestations. There's early Lyme disease and chronic Lyme disease, as well as disseminated Lyme disease. As of now, there is also no clear diagnostic test, so it's very difficult to diagnose early. There is no human vaccine that's being developed, and the only treatment that's available as of now, if you catch, it really is di doxycycline, the well-known antibiotic. If you look at the picture on the top right, you will see reported cases of Lyme disease in the US in 2019. You can see it is specific towards coastal regions, and then underneath we have a picture of exotic scapularis tick. Another really important factor about Lyme disease that we have to consider, especially in the context of this presentation, is the life cycle and potential host reservoir hosts that Lyme disease can live in. And in fact, so Lyme disease is able to persist thanks to an enzootic life cycle, so that the spirituary, the tick, and then most of the time a mammalian reservoir host, but it can also extend to reptiles and birds. There's four main stages of the tick life cycle, going from an egg to larva to nymph and adult, between larva and nymph and adult. There's to be a two blood meals taken from any of those reservoir hosts that we talked before, so if it takes a blood meal from an already infected reservoir host, it will then take up to spirituaries and be able to transmit it to another host. So, or if it bites to a human, that's now humans would get infected with Lyme disease. There is no vertical transmission, so it has to feed off it infected host. So this also kind of play into the whole point of this paper, and it really focuses on what factors of host take inspire key interactions affect the prevalence of Lyme disease infections throughout the US. So specifically in this paper, we are going to be looking at the differences between the north and the south of on the east coast. So the paper title is Why Lyme Disease is Common in the Northern US but Rare in South, the Rules of Host Choice, Host Seeking Behavior, and Take Density. It is published in early 2021. So it's a very recent article, and you can see the authors that are listed here that collaborated on this project. So some more background for this paper. If we look at the top right figure and panel, you can see exotic scapularis distribution. So where it could those takes be that are holding these spirituaries that could give human Lyme disease. So you can see a lot of density in red. There's the exotic scapular ticks, mainly in New England, Florida, and it gets less dense towards Virginia, Kentucky, and the northeastern parts of Georgia, Tennessee, and North Carolina. So then, if we look at panel B, you can see it takes that are you can see it takes that are actually infected with Borrelia, and prevalence is just drastically different in the northern states than panels. Then panel we can also see human cases of Lyme disease. So underneath that we have the reservoir competence. So how likely are these reservoirs going to be able to transmit Lyme? And we can see it's mainly successful in small mammals. So from all this, from their beginning figure of this paper, we can see we can see that human Lyme disease is mainly in the New England, and there is a ton of different reservoir hosts that are possible. But why the question that there are why the question that they are trying to ask is why it is more prevalent in North than it is in South. So to get into how they are gonna answer this question, we have to look at the methods that they used. So their sampling program included 9 sample sites that we can actually see in the previous slide, which included Wisconsin, Massachusetts, Rhode Island, New Jersey, North Carolina, Tennessee, South Carolina, Alabama, and Florida. We had live traps that are we have live traps, and they collected wildlife for 2 nights, and they also collected ticks every 15 meters surrounding the trap. This paper also did a really good job describing their estimation methods and their statistical analysis. So for their estimation method, they use regression, the Shannon Diversity Index, a logistic program procedure, and disparage estimation program, all very known. And then further infection testing, that is where we get into the molecular method part. They used DNA that was extracted from collected ticks from around and trap sites, and they use 16 single 16S ribosomal RNA qPCR, and they use specific probe for Borrelia burgdorferi to check and see if those ticks were infected with Lyme disease. Spark heats for their statistical analysis. They use GLM procedure and program called SAS, and they also accounted for variables that could have made a difference in their analysis, like year, site, host abundance, take abundance, and few more that they have within their paper. Now we are gonna get into a few of these figures. So for figure 2, they have a lot of different measurements between the north and the south to see if there is any statistical significance between host diversity and tick abundance. So panel A is gonna be your Shannon index. For trap species B is your richness overall of the 7 sites. C is the evenness. D is the species richness of the number of species of animals that were photographed. So instead of collected, and then E is the larva, F is the nymphs in 2011, G is the larva in 2011, and H is the name in 2012. So from this you can kind of see thankfully they have a statistics on every single one of these graphs. You can kind of see where there is a positive trend, negative trend based on the latitude, so N versus south, and it looks like the larva per sample is significant where we were looking at the latitude for both 2011 and 2012. So they have found a significant difference is in the amount of larvae, and they were on the host that were collected at the trap sites. Figure 3 is the proportions of the host of different categories in samples and proportions of ticks collected from host in each category. And the main thing to get from this figure is if you look down at the bottom, you're gonna see all of the different types of potential reservoirs hosts that were looking into how much larvae and nymphs are on them. So you will see the most of the southern states, it normally stays more towards reptiles for both larvae and names, for both larvae and names. As then you get into more different northern states, it's a small mammal. So another difference is the host selection. Figure 4 is the latitudinal trends and tick host associations. So you can have odds ratio similarity between latitudes, and also they use 95% confidence in limits, and they perform statistical significance tests like they did in other two panels well. And by looking at the similarity difference, this is the kind of where they were trying to prove, okay, is the dilution ticks throughout the north and the south. The main reason why there is difference in Lyme disease, or it based on latitude, so we would have to see a perfectly similar trend if it was about tick dispersion. But we are seeing is there is a similar amount of ticks, but there's a very descriptive latitude difference if you look at the panel C and D for larvae and nymphs. So they're looking at its as is this ticks distribution reservoir host association, or is there something else based on the north and south. So this is the next panel that we are looking at, is their fifth figure, as they're looking at latitudinal trends and ticks number on host. So they are looking at the mean number of ticks per individual animal in each category over the season. So this is again kind of looking at mice, voles, skinks, the small mammals versus reptiles. As you can continue to see the association between south versus north, small mammals in the north, reptiles on the south. And then the next table that there is the factors affecting the number of ticks per animal host. So you can see they looked at ticks abundance, host species richness to see if there is any significant difference. And you can see in the south they found significant differences from skinks, and the north there are significant difference from mice, and for larvae, and then for nymph skinks there are significantly differences in the south. So they kind of sum everything up in this panel that we have here, and this is where the main epidemiology emphasis is. So between all these associations that they form, the main thing that's important in which one are infected and where they are infected. So that's where we get into molecular methods of qPCR with 16S ribosomal RNA. So along the horizontal axis, as you can see, most of the northeastern states along with Wisconsin and have higher amount of infection rates versus the southeastern states. Almost besides New Jersey have much lower level of nymphs, larvae, and reservoir hosts that are infected. This kind of shows the whole point of this paper. So what they are getting at is if you get bit in the south, since the larvae and nymphs that are biting the reservoir hosts are statistically less infectious than in the north, humans are going to be less likely to be infected, as at least that's what they are implying from this data. So to sum it up, I thought it might be a good thing to pull it up. Sentence that I thought brought everything together in this paper is the sentence that I thought it would be a good one, where it says uh, north-south trend in tick infection prevalence with Borrelia burgdorferi, burgdorferi, and the gradient of human Lyme disease cases throughout the U.S. is from the selective attachments of ticks to lizards, mainly in skinks, mainly skinks in the south. Um, they also found that the tick abundance and the dilution effect did not explain the difference in Lyme disease cases in the north and the south. So that was our first results, like slide looking at is there is anything specific about where the ticks are or why there is more in north versus the south. They also found that the lymph nymphs they also found that the larvae and nymphs are more active in the north during the same season, so that could have attributed to an increased amount of infection. They found that there was two factors affecting infection rates. So mainly, there's less people that are bit by nymph ticks in the south, and less ticks are infected in the south, which could mean that there is less Lyme disease cases. So these are just the main points that they discuss from their data, and nearly already known data about Lyme disease and tick distribution is really what's interesting, as we're gonna talk about in the next slide. So the major epidemiological implications from this study that they have found are some major things: that there is difference between the type of host that ticks will latch into based on the north versus the south, and there is just generally less infectious species found in the south. But if you look at the map, the forecasted prevalence of Lyme disease in 2022, we can see the infection rate is moving further south, further west, and it should increase significantly over the next year. Another thing that they could have done better is larger sample size, more sampling areas, that they could have done more than just 16S ribosomal RNA from their molecular method. It would be interested to see them explaining an association between human Lyme disease cases and how those Lyme disease cases were confirmed, and overall Lyme disease has just become a silent epidemic over the last 40 years. And there is also a need for a big push for treatment, diagnostic technology development, and human vaccine would be extremely helpful, because this disease can be super debilitating. So I would like to thank everybody who watched this presentation, and these are my references. And again, thank you very much for listening to this presentation. I hope you liked it. Thank you very much.